Okay, cool. We are starting to stream on YouTube. We are recording. Okay, cool. We are starting to stream on YouTube.
So Gilad, let's wait another minute.
Yeah, we can wait.
Is there still people joining?
Yes.
So I think it leveled off by now.
Yeah, I will continue.
Actually, let's not steal too much time from Ken's talk.
Yes.
Okay, so welcome everybody. It's wonderful to again see so many of you for our third webinar on protein folding and dynamics. I just want to briefly mention before I introduce our speaker today that in case you encounter problems in joining Zoom, there will always be the chance to follow the seminars on YouTube, on our YouTube channel, either using the live stream or also later on by listening to the recordings. I will just quickly share my screen with you. So you find the YouTube link on our website, proteinfoldinganddynamics.com. And on that website, you will also find a list of all the upcoming talks with titles and abstracts. So check it on a regular basis in case you're interested. Now, our speaker today is Ken Dill, and it's really fantastic to have you with us, Ken. Thanks so much. I don't think that Ken requires a long introduction since he's well known to all of you, but for those who are not familiar with his work, Ken is the director of the Laufer Center for Physical and Quantitative Biology at Stony Brook University. But his group is also affiliated to the departments of chemistry, physics, and astronomy, which already tells you that his research program is extremely multidisciplinary. His interests range from protein physics, where he's done breakthrough work in using statistical mechanics concepts to describe protein folding processes, over cell behavior and up to extremely fundamental aspects such as water behavior. Now, Ken is a member of the National Academy of Sciences, and he received a large number of honors and distinctions. And I will just mention the more recent ones. Ken received the Max Dierbrook Prize for Biological Physics last year, and also the Sackler International Prize in Biophysics in Tel Aviv, actually not far away from the Weizmann Institute. He was also elected a member of the American Academy of Arts and Sciences. And I would like to mention that among undergraduate students, he may be well known for two excellent books that he wrote. He's not only a fantastic scientist, but also an excellent teacher. He is one of them, Molecular Driving Forces. In my group, it actually became the standard and simply called the book by now. Now, Ken will talk today on how forces on proteomes manifest as cell behavior, so a sort of synthesis of protein physics and cellular behavior. But before I hand the virtual stage over to Ken, I would like to ask each of you to please mute your microphones such that we can listen to the seminar without interruptions and background noises. As usual, the seminar will be followed by a questions and answers section of about 15 to 20 minutes. In case you have a question during the talk, don't ask it immediately, but just write your question in the chat window or simply write in there, I have a question, and I will call you after the talk one by one and you can ask your question in person. Now, without further ado, I would like to hand over to Ken. I stop sharing my screen and ask Ken to share his. And so thanks again, Ken, for joining our webinar. And yeah, we are very much looking forward to your talk.
First of all, it's great to see you all. And I want to say thank you to Hagen and Gilad and Ben for setting up this great idea, setting up this wonderful community, inviting some terrific speakers in the past couple of weeks, and executing it beautifully. It's been just fantastic. I also appreciate the fact that we have here in the world's craziness, hopefully a few minutes of respite where we can sit and chat together about some science. All right, so what I want to tell you about, let's see if this is going to work now. Can you all see that?
Yes.
Okay.
Yes.
Okay, great. So what I want to tell you about is some work we've been doing in the last few years at Stony Brook. The question is about cell adaptation, which I'll define in a moment. And the issue is the mechanisms that are associated with proteome physics, and particularly the forces. So let me first start by defining what I'm talking about. Homeostasis and evolution are the two forms of adaptation. I'm thinking about what I mean by adaptation then is if I take a cell and I change the temperature of its environment to something else, the cell may behave differently. We know this through growth laws that are shown in the bottom figures, for example. And if I then perturb back to the original temperature, the cell will have the same behavior it had. This resembles very much state functions of thermodynamics that are not pathway dependent, but they're dependent on the environment. And so it's that dependence on environment and what it is that cells are trying to do in different environments that we're interested in. Homeostasis is applicable to individual cells over their lifetime or short times, and evolution is applicable to populations that aim to find fitness peaks on fitness landscapes. And I show here three examples. The growth rate of E. coli as a function of salt, as a function of food like glucose, sugars, things like that, and as a function of temperature. I'm going to go into a couple of these in a little bit more detail, but these are some of the key quantities and properties that microbiologists care about. So for example, salt dependence is part of the reason that we put salt on pork to turn it into bacon, for example, and make cucumbers from pickles, and the temperature dependence is why we have refrigerators and stoves. If you cool things enough, what you see is the growth rate there. I can point to this. Let's see. If you cool things enough, the growth rate goes down. If you heat things enough, you kill them. So we sterilize, we have autoclaves, we have stoves, we have refrigerators over on this side, for example. And salt, we salt meats and fish and so forth to kill bacteria on them. So these are all well known. These growth laws are well known. I want to take a look at the role of the physics of the proteomes. One of my 30,000 foot points here is that we have three ways that cells can adapt that you know well. One is through a protein action. I can make a protein better or worse at the enzyme action that it performs, for example. That's this channel. Or I can take that protein and increase it or decrease its concentration. I can do that by changing the mRNA levels and change the expression. So that's what I can do with the protein, the individual protein level. But protein physics gives me a third channel of ways that cells can adapt. And that has to do with folding and aggregation and other ways that energies and forces and diffusion enter the problem in ways that don't depend on the protein's day job per se, but rather depend on universals of all proteins. So just a brief overview. In what way is proteophysical adaptation, the kind of thing I want to talk about here, is it different than adaptation from individual proteins and their expression levels? So first of all, we're dealing with sort of whole proteome level problems, not the protein day jobs, things like folding aggregation, synthesis, degradation, the diffusion rates of the proteome in the cell, and so on. Biology that we call persistent and not mutable, and what I mean by that is sort of more 30,000 foot biology, things like growth laws and aging and diseases of multi-molecules that aren't dependent on individual changes or individual mutations or amino acids, for example, that things that you can't mutate away. You can change the lifespan of an organism by a few mutations, but you can't change the fact that it ages or doesn't age. And you can't disappear all cancers with a few mutations. And likewise, growth laws, you can change a few of the parameters, but you can't change the general feature of it. So these are the levels of things we're interested in. Briefly, I'm going to give you about 4 or 5 vignettes. David Eisenberg years ago made a very useful point, I thought, which was that when you give a talk, you have 2 possibilities. Either you can communicate 1 point or you can communicate 0 points. And so unfortunately, I'm going to try to communicate 5 points. But the reason that I'm doing that is because it's not the individual growth law or process that I want to get across to you. What I want to get across to you is a sort of a general approach to problems that we think might be useful also outside of the things that I'm telling you about. So I want to give you sort of an overview picture of all that. So here's how we approach problems. First of all, we think about what environmental variable do we care about. We hypothesize what degrees of freedom might matter. And in this case, we're not talking about genomics from genotype to phenotype. We're talking about degrees of freedom that are a little higher level than this and a little more complicated to understand from amino acid sequence, but nevertheless are the things that the organism can work on. Then we hypothesize a fitness function, and we look for the peaks in fitness function to figure out what the cell is trying to do. So these are the kinds of pictures, fitness landscapes, where the basic idea is in evolution, cells are trying to, populations are ultimately trying to reach to peaks of fitness landscapes. And in homeostasis, we have a similar kind of function where you have something like a minimum that the cell is trying to achieve, like a minimum of an energy function, except it's a homeostasis function. So let me start on the first one, the salt growth laws. So how does external salt affect growth rate? I'll show you real data in a minute, but I just want to get the general point across that E. coli, for example, starts here. And the little red dots are showing you a salt concentration, and the circle is showing you, say, E. coli. So E. coli starts here, and if you add more and more salt, what happens is it slows the growth. And I'm showing you also on the x-axis a hypothesis about a mechanism. So this is where our hypotheses come in. What are the degrees of freedom? And ultimately, what is the fitness coming from? And the hypothesis here is that a key driving force is the osmotic pressure. So as I add more salt, I've got more red dots. What happens is the cell will shrink because it's drawing the water out from normal osmotic forces. And the shrinking causes more protein crowding inside the cell. So the mechanism we hypothesize is this, that it's the crowding of proteins inside the cell that is the reason for slowed growth, and it's because the proteins can't move as well. We call it the protein mingling rate. It depends on the protein density, and if you make a cell too dense, you have too much protein and the proteins can't move around. You're all probably familiar with these beautiful David Goodsell pictures. He's trying to show us here sort of anatomically correct pictures of a cell in the sense of the crowding of proteins. The cell by volume is about 20% protein. It's pretty crowded. If you were thinking of this as colloid physics, that would be so crowded that things would precipitate. But for various reasons, they don't really precipitate inside a cell. The basic argument here is we have two opposing forces that on the one hand, so let me say a word about what is this mingling rate. Mingling rate is, think about lysozyme, for example. If I need lysozyme to be able to make it throughout the cell fast enough and to be able to interact with all the other proteins in order to get protein-protein interactions, I need enough concentration of lysozyme, but I also don't want it too crowded. I don't want the whole cell too crowded. So those are the opposing forces that we hypothesize. If it's too sparse, I don't have very much traffic because I don't have very much lysozyme. If it's too crowded, then the proteins move sluggishly. We can make a very simple model of this. All of these, by the way, are really simple physics, so it's at the level of kind of 6th grade physics or something. What we're looking to do here is to compute, think of proteins as spheres inside E. coli. They're diffusing around, but they're diffusing in a crowd of other proteins. So the mingling rate, just from well-known kinetics, is the concentration times the diffusion rate of that protein. And so the concentration C is here, concentration of total protein of all types. And the diffusion constant is the normal diffusion constant, Stokes' law, 1 over the radius A. But now I have this extra term, the 1 minus C squared term. That's the first order colloid theory for what happens in a crowded medium, how motion slows down in a crowd. Now, all we do here is we say this is the fitness function. The fitness function for the cell, the salt growth for a cell, is simply proportional to how fast the proteins can mingle, which is given by this function of concentration, which depends on the osmotic pressure. And I now can look for the peak by taking the derivative of this fitness function with respect to concentration. I'm asking now, how dense does the cell want to be? And the answer from this, plugging in numbers for an average protein size inside E. coli, it gives you C star, which is the peak point, is 20% crowding. So that's interesting, but it's only a single number. But the single number is telling us that the crowding level of proteins inside E. coli does happen to be consistent with this simple little picture. But we can go beyond and also ask, what does that C times 1 minus C squared function look like? And it looks like this. So the two curves, forget about the difference between them. They're just difference in choices we make about which proteins might matter. Is it big ones or is it all of them averaged? And it turns out it really doesn't make much difference. And then here's data taken from Tom Record's lab a few years ago as a function of the volume fraction, which he converts from having used osmotic pressure to be able to change the density in E. coli. And this is a validation of an aspect of the model, which is to say, this was Weishar with Tom Record's group, both at Wisconsin, did an interesting experiment of taking green fluorescent protein and asking what happens if you change the osmotic pressure outside, how fast does GFP move when it's inside E. coli? The x-axis is a log scale. So you see 2 orders of magnitude difference in diffusion constant. Here's the outside external osmotic pressure, and the osmotic pressure is going up here, which means higher salt concentrations. And what you see is the diffusion constant of GFP drops by 2 orders of magnitude with a 5-fold increase of salt concentration outside. It's simply, the argument here I'm making is simply that this experimental data is consistent with the idea that it may well be the protein sluggishness that's the basis for this growth law. Very simple argument. Next example is food growth laws. This picture is an example of one that's probably one of the best known experiments in all of microbiology, which is you put certain amounts of glucose or various sugars outside of cells, like E. coli in this case, and they grow at different rates. The more food, if you start down here with small amounts of food, the more food you add, the faster it grows, and then it plateaus. You get to a certain speed limit where more food no longer, the cells can no longer grow any faster. They're running at their speed limit. This is a picture from Minot back in the 1940s, and it's a very common experiment these days. The question is, can we look at a mechanism at about the same level of coarse graining as we did just a second ago for salt? What is the mechanism for this kind of curve? And in particular, I want to raise an issue about what dictates the level of the speed limit. There is a puzzle here. Here's the puzzle about the speed limit. The question is, why can't E. coli duplicate in 6 minutes? E. coli duplication speeds, when everything is provided for E. coli, amino acids, glucose, everything you want. When you give E. coli the full-blown best conditions it can have, it never duplicates any faster than about 20 minutes, 25 minutes, something like that. There is a factor of 3 because here's a little plot. First of all, we know that growth speed is proportional to the number of ribosomes. That makes a lot of sense. If I want to duplicate faster, if I'm E. coli and I want to duplicate faster, I just turn more of my ATP energy into producing ribosomal proteins relative to non-ribosomal proteins, and that way I can parallel process to grow a daughter cell by producing a lot of protein as fast as possible. So there's a sort of a physical principle speed limit, but this is one that E. coli never gets to in real life. And that speed limit would be if the fraction of all the proteins produced in E. coli were just ribosomes. E. coli is just a bag of ribosomes. Then it turns out every ribosome only needs to make 1 copy of itself, and then you've produced a daughter cell. That would be the speed limit. And that would be about 10 per hour, and that's about 6 minutes. And that's just taken by how fast I add amino acids onto protein chains and how many protein chains do I have in a ribosome and so forth. So 6 minutes is the absolute speed limit in principle, for how fast E. coli could duplicate. But in reality, what you get is this red dot. First of all, it's about 1/3 of that speed. And secondly, the fraction of ribosomes is not equal to 1. So this is the question. Can we make any sense out of those numbers? And so what I want to argue is that just thinking of E. coli as a little NASCAR built for nothing but speed misses the point. And it's why we think there is this discrepancy between this number and this number. And sort of as tangential evidence that biology is not always out for speed, here we have a cow, which is clearly not built for speed, and we have a tree, which is clearly not built for speed. And rather, this picture shows you something interesting, I think, that surface to volume ratio matters enormously depending on whether you're mobile, like a cow is, like we are, like fish and birds are. If you're mobile, you have to carry your fuel with you. On the other hand, if you're immobile, like a tree or a bush, for example, and you're planted in the ground and you get all your energy from sunlight, you have a very different energy life than you have if you're a cow. A cow needs to carry fuel, so it needs high energy density fuel. It needs surface to volume ratio that's pretty spherical. And this in principle is where I'm supposed to go into a spherical cow joke, I think, but I'm not going to stoop to that. Anyway, you want surface to volume ratio here to be very small, and you want, because of dissipation and how much stuff you need to carry, here you want surface to volume ratio to be huge. And you can see it is what biology does. It cares about energy a lot. And so we took that page and here's our model. It's the simplest model that I know to make. Here's E. coli. It's fundamentally about energy, the energy balance, and how the energy gets trafficked, if you will, to ribosomes versus proteins. Glucose comes in the front door, makes ATP. ATP then can make ribosomes, and ribosomes are needed to make non-ribosomal proteins, or ATP can go to making proteins, and proteins are what help me convert glucose into ATP. So everything's talking to everything. These are nonlinear equations, but there's only 3 of them. I have one for the ribosomal protein concentration, one for the non-ribosomal protein concentration, one for the ATP concentration, and I have one that's just a conservation of all the protein mass. It's the sum of these 2 pictures. So what I can ask now, these are nonlinear equations, but it's in steady state. I set these quantities to 0. So I have 3 third-order equations. I can solve them, and then I get a fitness landscape. And what I do with this fitness landscape is I say, well, okay, I already know that speed is not the whole story for fitness. Maybe it's energy efficiency. So energy efficiency we define here as growth rate divided by the ATP burn rate. This way I avoid issues about oxphos versus fermentation and so on. I'm interested in just per ATP, what is the growth rate? And again, this is my fitness function. And again, I take a derivative, I set it to 0, I ask where's the maximum. Turns out you can get an analytical result from this. I won't go into the details, but let me just show you the answer. And the answer is, this is what the fitness function looks like when you calculate it from this 3rd-order polynomial model. On the x-axis is the fraction of all proteins that happen to be ribosomal proteins. We have only 2 choices, being ribosomal protein or non-ribosomal. This is the fraction that are ribosomal. And on the y-axis is the growth rate per ATP. And our prediction from taking the peak point of that curve I mentioned before is 0.8. And what you see is experimental data on E. coli under all kinds of different growth conditions and different food and stuff like that. Peaks around 0.7-ish, somewhere in there. So not terribly far off, but also you could say, well, maybe we can do better. Now, to determine what the model is trying to tell us, let's go back and ask, why are we getting this peak point? What's the physics of the peak point? And the answer is, it's very simple. If you think about a NASCAR or race car machine that I want to make really fast, but I'm also thinking about something that I want to make pretty efficient too. I have a carburetor front end, which is the thing that in this case, lets the glucose turn into ATP, and I have a motor back end. And it turns out if I have a very big carburetor and a very small motor, first of all, it doesn't grow very fast, but also it's not very efficient. If I have a small carburetor and a big motor, that's also very inefficient. But if I match them, the carburetor is the right size to run the motor, that's basically where this peak point is coming from. So it says, if I'm E. coli, it can't be all motor. I have to have enough proteins that convert glucose to ATP. Next vignette has to do with biomachines, molecular machines. This is showing you a picture of FOF1 ATPase, a little toy cartoon of it. What is being optimized here? Machines have all kinds of things going on. They have speed, they have efficiency, they have force, they have work, they have power, which is the production rate. Which one of these, if any, might be the ones that molecular motors and ion pumps and so forth optimize? Well, so first, if you look at the data, it's very interesting. Many biomolecular machines are very, very efficient. Here's the input, let's say ATP conversion to ADP. Sometimes it comes from gradients, but anyway, this is the input, and this is the output work of machines that do work. And here, this black line is showing you 100% efficiency. Nothing can be higher than that, but you see a few motors along here that are very, very close to 100% efficient. Now, some people tend to think, well, gee, doesn't that violate thermodynamics? And the answer is no. The Carnot limit is only for thermal machines. It's only for heat engines. And biological machines are all chemical potential engines. So they aren't subject to the Carnot limit. So there's no problem in violating anything here. But what we are interested in is, well, okay, how do they do it though? How is it that biomolecular machines, some of them can be very efficient, but also there's some that aren't very efficient at all. And the question is, are there deeper insights we should take advantage of? So this shows you speed versus efficiency. It shows you some machines are terribly inefficient, like RNA polymerase. It shows you other machines are very, very efficient, like FOF1 ATPase. That's a good deal because this is producing something like half of the energy inside of all of us, and it's a great idea to be pretty efficient. So how do we model this? So this is work of Jason Wagner in the group. He's made a beautiful, very simple little kinetic model, which says that motors have a front end and a back end. The front end is the chemical intake, let's say, of ATP, and the back end is the mechanical output, let's say, producing work. Now, we can fool, this is just a little phenomenological model. We can fool with the barrier heights all we want to, and the knobs on those barrier heights are our parameters, lambda and delta, which allow us to explore how to optimize a machine, a molecular machine. And you can see here, on the one hand, I could have all of the barrier in a Brownian ratchet, I have all of the barrier in the second hill. Whereas if I have something like a power stroke, which we call a driven motor, instead, then I'm distributing the barriers so that I have about equal barriers in the front and the back. And it's interestingly, in a power stroke motor, you get way more efficiency for speed. So this is a great idea. In a Brownian ratchet, you get very little. And what this figure is showing is that the blue curve is showing you that the way you can distribute these barriers, the way you can get this power stroke, is you have to store energy somehow in the middle. And you want that energy storage to happen in the early stage, the chemistry stage. I'm not going to focus long on this, but just to say that what we can toy around with a little model that allows us to understand it at a very coarse-grained level. My proton is going to go through here. In essence, let me just say, since I'm going to be running short of time in a minute, let me just say the essence is the proton wants to flow down its gradient and would do so at a particular speed. But by having a rotor in the middle that can store energy in twist, it turns out I can slow that proton down. And that's a good idea, because what I want to do is slow down the input step so that I can speed up the output step. And that's what it does. It stores speed. We're moving a barrier around. And so there are ways you can do this by having a simple rod that both twists and stores elastic energy and rotates. Any way you can make cycles. I don't want to go in more detail on that. And this point shows that in addition to how we could change the internal parameters, in evolution, we have other problems too. The lambda and the delta I mentioned already are our internal parameters. That's what I just described. But now I'm going to fix those and look at these 12 machines I looked at before. And I'm going to ask, to meet sort of the performance needs of the environment, I have a particular kind of input I want to be able to use if I'm trying to design a motor. Maybe I have ATP that's going to convert. That's my input. And maybe my output is I have to perform some kind of a job, some kind of work. And so the question is now, based on the performance characteristics, what input am I going to get and what work do I need to do? The question is, what is the optimization, the fitness function? And what we show in this curve for motors and in this curve for pumps, ion pumps, is that the fitness function seems to be speed times efficiency. It's maximizing the power. It's both a sort of a force and a speed thing. Okay, the kind of the last story I want to tell is about cell stress response and how proteostasis decisions are made by using ATP. Can I make decision machines instead of just workhorse machines? So the gray panel here is showing you normal folding, native up here. That's where you want to be. Unfolded, misfolded, nucleus of an aggregate, and then full aggregate. This is the E. coli diagram of the main chaperones. So this is DNA KJ, CLIP-B, the LON protease, trigger factor, and GRO-EL. You see there's a lot of trafficking here. What we're trying to do in this problem is, this is a bigger challenge than the ones before. It's not just a sort of single variable thing. It's a whole dynamical network of rates, steady state rates. And so we know lots of this thanks to a lot of the people here in the audience and others in the biophysical community who've measured all these trafficking rates, for example, of unfolded proteins or misfolded proteins that enter DNA KJ or that enter GRO-EL. We also know some things about the folding and misfolding parameters themselves, rates of folding and unfolding and so on. So we can build a big matrix from data, and then we can ask questions about how is this machine designed and how does it make decisions? And we think of it as like a hospital for sick proteins. It's you in a hospital, you're a patient, you walk into the hospital, you have to let the hospital know what your sickness is. So different proteins misfold in different ways and to different degrees. Second, in the hospital, the patient gets sent to the right doctor, and then finally the doctor fixes the problem. This is a decision machine in a hospital. This is what it looks like for us. We have these parameters from data. This is what the answers look like. It turns out, so Ulrich Hartl has these different classifications of protein health, if you will. Proteins he calls class 1 proteins are ones that fold pretty well by themselves. Class 2 are a little sicker and need some help. And class 3 are really sick. And it turns out the really sick proteins all get channeled through the GRO-EL system. The less sick proteins get channeled mostly through the DNA KJ system. And the proteins that are reasonably happy on their own get channeled mostly through trigger factor, which doesn't cost any energy. It turns out if you stare at this a while, what you see are some very sensible decisions. The sickest proteins go to the most expensive chaperones, for example. Time doesn't permit me to go into a lot of detail here, but I just want to say that when you slice and dice this sort of 27-dimensional machine, if you will. By the way, a lot of the parameters for this machine are known from independent experiments. This is not a curve fitting exercise mostly, there are a few. But it's a pretty clever machine. First of all, it identifies the proteins, how unfolded or misfolded is the protein, sorts it through the right chaperone system. It's an unmisfoldase, so it acts on the misfolded states, not on U, not on N, not on aggregate. And some operate in different places, but largely it's an unmisfoldase. It acts on client misfolded states. How long is a molecule dwelling in the misfolded state? That time constant dictates who swallows it up, which chaperone. It's energy efficient. The sickest proteins use the most expensive doctors, if you will, or chaperones. And that turns out to be not a choice of the doctors, it's a choice made by the proteins. The sickest proteins have the tightest binding affinity for GRO-EL. It's got a good balance of other things that also adapts to growth and stress. High growth and low growth are both stresses, and they need upregulated chaperones. So my final point, I know I need to end here in just a minute. My final point has to do with what happens when the cell is maladapted. And here we focus on proteostasis collapse. There are many things that are problems when as cells get old and die. we focus on oxidative damage. Oxidative damage hits pretty much everything. It hits DNA, it hits lipids, it hits proteins. But I'm going to focus here on proteostasis collapse. In part, a lot is understood about that. And in part, we do think that it's at least a dominant factor in cell aging and death. And it does give us a picture of what happens when cell adaptation is no longer working appropriately. So here's the evidence for why this proteasome collapse and proteostasis collapse is relevant in aging and death and disease, and aging-related diseases. And that is, of course, aging is a major stress, and proteostasis is the front line of the stress response system. It's been noted in the literature that all gerontogenes, all aging-related genes, are stress-related. The main risk factor for the 15 proteinopathy diseases is age. And so there's various things. And also one point, let me make this point, that aging is not about one gene or another. You certainly can modulate it. Certain diseases, certain genes like the DAF genes, for example, Cynthia Kenyon knows, you can really modulate lifespan by quite a lot. On the other hand, the aging process itself, you can't disappear. And so aging has to be something that's bigger than a single gene or a single pathway. And so we think that that proteostasis is where a lot of the action happens. This point is that if you look at all kinds of different organisms and you normalize an x-axis of their lifespan, so humans have lifespans of say 90-ish years, and worms are much shorter. C. elegans, for example, is about 20 days. You normalize their lifespan and you plot the carbonyls, this is an indication of protein oxidation. What you see is they all fall along a kind of a universal curve, that for the first half or more of your life, your oxidative damage is being controlled by various repair. But near the end of the life, what happens is it gets out of control. And this red band here is indicating this human disease called progeria. This is where people who are like 16 years old look like they're 80, for example. And they have very high oxidative damage levels even when they're young. So we take the model I just described to you a minute ago, our proteostasis model, and that is, this top plane is exactly the model I mentioned before, except we strip it down something to its basics. We don't have all 4 chaperone systems anymore. We have one average chaperone system. We have one average type of protein, and we just have a few states of proteins folding. And so we simplify, but we keep basically the essence of what we had before. But now also I want to go beyond E. coli, and I want to go to C. elegans. So it turns out the one thing that you have to do with all those parameters we got from E. coli before, all of the traffic parameters of the binding affinities of this protein to that chaperone and so on, is to reduce the speed by a factor of 10. And then otherwise you can take the same set of parameters. Now, so this is what the model I just described before of a healthy young cell that's growing and has normal chaperoning. Now, our damage model for aging is that oxidation happens at a slow rate over the lifetime of the organism, and that damage is irreversible. Once you screw up a protein with an oxidative damage event, now this is a fraction of protein, some oxidative damage is not so harmful. Once I screw up a protein with an oxidative damage event, I'm stuck in this damage plane and I can never get out. And so if I can then, I can add up all of my misfolded and native proteins, and what you see is over time, I'm going to lose some native proteins. So it's the same model as before, the hospital model of chaperones, but I have linear oxidative damage with age over a long lifetime. And some damage is irreversible, and these parameters are the same as the ones up here otherwise. What does it predict? So I'm down near the bottom here. There's actually some great biophysical data in C. elegans. This one is stunning. The data is stunning. If you grow worms at 20 degrees C, which tends to be the sort of the normal thing in aging studies on C. elegans, then the lifespan is about 20 days. On the other hand, if you grow worms only 15 degrees hotter than that, the average lifespan over the whole worm population is 1/100th of that. It's like taking people and saying, if I moved your family to the tropics, then instead of having a 100-year lifespan, you would all have just a 1-year lifespan. It's really stunning. This is the biophysical data. And it turns out this is what the model is. We know the temperature dependencies of folding and the trafficking and so on, and that's gone into our hospital model. Similarly, oxidative damage is augmented. You can put in these this T-butyl peroxide compound at increasing concentrations in C. elegans. And similarly, when you put that in at higher concentrations, the lifespan goes down by 2 orders of magnitude as well. There's a famous Gompertz law. The older you get, the higher the probability next year you're going to die. And also chaperones can increase the lifespan, although there's counterexamples, by the way, to that. And the lines there were showing you the predictions. This is showing you a survival curve just to sort of put the lifespan on an x-axis. Let me just summarize this briefly. What it tells us about, what the model is telling us about what the tipping point is when aging turns to death is you get this peak of misfolded protein, and then you overflow the chaperones, and then the aggregation starts to run wild. And what happens is you form so many bad proteins, so many oxidatively damaged, irreversibly damaged proteins, that the chaperones are totally distracted trying to fix problems they're never going to be able to fix. And that's what happens, is your chaperones just spend all their time in the wrong place. Summarizing all of this, if you thought about a cell with no synthesis degradation or chaperoning, you would have something like an average concentration of good proteins is like a lake level, but over time they will misfold and they will aggregate. And so what will happen is your lake of good proteins turns into destroyed proteins. But in real cells, of course, you have 3 processes. One is new proteins are synthesized, which fills up your lake of good proteins. One is you have chaperones, which turn misfolded proteins into well-folded, and your bad proteins are degraded. So this keeps your lake level of good proteins high when you're young and when you're healthy. What happens in old cells, and when you reach the tipping point, you end up getting slower protein synthesis. That's outside our model. That has to do with energy, ATP levels, and so forth. You get slower degradation, and the chaperones now are also so overloaded they're not functioning very well, and oxidative damage is the driving force that basically drains the whole swamp here. Summarizing it all, just in a nutshell, the argument is that we have some fairly simple models where what we do is we assume or assert certain degrees of freedom and certain fitness functions, and then we ask, what can we predict that way? And so for salt, it's about protein diffusion speed. For food, the upswitching of growth speed is an upswitching of ribosomes. And this is about maximizing energy efficiency. Motors, what we learn is that if you want to design, if your evolution, you want to design a motor, you should be sharing the speed across multiple barriers if you can. That gives you much more efficiency. Proteostasis spends ATP to build a decision machine that turns out to be a pretty smart one. And in aging, the point is you've exceeded all your adaptive capacity at that point. Finally, these are the folks that have done a lot of the work, and these are a lot of students and postdocs and friends and associates that have been a wonderful community to work with all these years. And thank you very much for listening.
Excellent. Thanks so much, Ken. Fantastic talk. And we already have the first question by David Nesbitt. David, do you want to ask a question?
Ken, thank you for your, as usual, just incredibly elegant, beautiful, simple talk. I love it.
Thank you.
And I'm going to respond with a simple question. When I go back to your SALT model,
Yes,
where you basically predict this 20% value, I think it was,
yes,
and it really came basically from the simple calculus of C star times 1 minus C star squared,
right.
the maximum in that fitness function. Is there a physical picture for the squareness of the 1 minus C star that I should be aware of and think about?
It's super simple. It's just that in colloid theory, what you get for the density dependence of diffusion of one object in amongst a bunch of other objects, it's a polynomial in concentration, and it's just the first order term is a C-squared term. That's all. It's just I'm bumping into stuff.
And why is the linear term not the first term?
Oh, sorry. There is a linear term. So it's C times 1 minus C squared.
Okay. Yeah.
Okay.
All right. So yeah, but I guess I was thinking in the 1 minus C quantity squared, you say that comes from standard colloid theory. And I'm wondering, why is there not a linear term in that Taylor series expansion?
So there is. Let me just see if, hang on one sec. Let me show you my screen again. Oh, I am, okay. So
Yeah, you are still sharing.
are you see this? Are you seeing this now?
Yeah, I am.
Okay.
Right.
Yeah. So this is the term. So you do have inside here. I think what you're asking, if I'm not mistaken, is inside here. Yes, there is a C. There is a C squared term. There's this C squared term. Okay, and this C is coming from the concentration of the protein itself, and then this term is just 1 minus C, but it's squared.
And what I was asking really is what makes that quantity the square, and presumably that means it's some first order Taylor series disappears.
Yeah, exactly. That's right. That's a good question, and I'll have to dig into that and get back to you.
Okay.
But,
Beautiful talk.
why it doesn't look like just a C squared instead of the quantity C. Yeah, good
but
question.
the square is on the outside, Ken. This is Ned.
Yeah.
I mean, when you square that, you do get a linear term.
Yeah, you do get a linear term. That's right, exactly.
Okay, next question is by Stephen Fried.
Hi, yeah, thank you so much, Ken, for a lovely talk. And I'm thinking that maybe my question will possibly also hit on what Catherine was asking about in the chat as well, which is that you alluded to that there might be some advantages for certain machines to be more efficient and other machines to be less efficient. And I'm not sure if you had a chance to sort of fully address that. And I guess, the simple reason that would come to me is that, clearly one of the reasons for having an inefficient machine is simply that it's just going to be more irreversible, right? Clearly, an RNA polymerase that goes backwards will be really bad. So you want to have to burn a bit of heat so that you really have, a very low probability of going backwards. Whereas an ATP synthase that every once in a while accidentally burns ATP to sort of pump protons, probably not in the world, considering there are other proteins that do exactly that. So I was wondering, did you have another insight or a more subtle insight, or was my sort of zeroth order thinking on that the same as yours?
Yeah. So here's how we're thinking about this. So the question, it's a great question, and it's why does RNAP sit on the left end with no efficiency and FO, F1 ATPase? Why is it so great? Here's what we think. It has a very different job. It is like a snowplow. It has to do a huge amount of work because what it's trying to do is melt the DNA double helix in front of it. And it needs to melt a couple of hydrogen bonds down the road. And so what I need basically is a little melting machine. And that melting machine has to do a huge amount of work, but the step sizes are very small because I'm just doing this base pair by base pair. So really what I need is a heavy lift, but I can work on a small level. Now the question though, I think that you're raising, which is super interesting, is, but okay, still, why couldn't I make that efficient in principle? And here's what I think at the moment, but we don't know, and that is that this mechanism that I showed you, a toy mechanism, is one where I can get elastic energy by twisting a rod inside the ATPase. and its conformational changes. And the question is, how big of free energy can I get out of an internal conformational change inside a protein? I don't know what the answer is, but what I think is that there's a limit to it. And that when I'm building a snowplow, I just can't store enough energy to make that happen, and therefore I have to do it inefficiently. And it's okay, I can get sort of decent speed. They're not very fast. RNA polymerase is not very fast, but it also doesn't need to be very fast because it only needs to run at the same rate as a ribosome. So I don't need speed out of it. I do need force and work out of it. And so that's we just think that the problem is somehow the protein design doesn't have the capacity to be able to store a large amount of energy in it, a sufficiently large amount of energy in it. That's all I can tell you. Hypothesis.
Yep,
Okay.
thanks.
So the next question by Catherine Roya. Catherine?
Thanks, Ken, but Stephen asked it and you answered it, so go to the next one.
Okay.
Yeah, so next one is by Kings.
Sorry, it was not a question. I just wanted to clarify the 1 minus C over C squared came from the diffusion term.
Thanks, Kings. Yeah.
All right. Okay, so next question by Raphael.
Okay, thank you for your insightful talk. So I wanted to ask, so you mentioned that some of the molecular motors are nearly 100% efficient, while there are motors which are less efficient. So what are the other general evolutionary principles for molecular motors, except for the increase in the efficiency or in speed times efficiency? Or maybe these motors that are less efficient, maybe they didn't have enough time for increasing their efficiency evolutionary, or yeah.
Well, so I can't say that I can completely outguess evolution here, but I can tell you a few things that Jason found as mechanisms that can provide one thing or another, speed or efficiency or work or power or what have you. One is, I mentioned already the business of sharing barrier heights. So storing speed, the idea of reducing one barrier height and increasing another one. So the limiting case of that is where when I'm building a motor, I have a whole bunch of stators and I can make many of them. So I have a choice of how many different stators can I make. And it turns out, so this is work that Jason did also that I didn't have time to show, but if you look at F1 ATPase, you look at a full set of proteins of having different numbers of stators in them. What you can see is that it does tend to be when you need efficiency, for whatever reasons evolution chooses that, you use more of these stators. And the reason you do that is because the more small barriers you can build relative to the fewer large barriers you build, the more efficiency you get. So you want lots of stators. And I think that's generally true with probably macroscopic motors as well. So there's various principles of that type. And there's principles about, do you want to perform the work in the forward step on the mechanical cycle, or do you want to perform the work on the backward step of the mechanical cycle? So there's a lot of those sort of mechanistic questions we can ask. What I don't think we have a handle on, certainly not a full handle on at the moment, is all the evolutionary why things are happening. It's interesting because if you look at different motors, I do suspect that there's more granularity and subtlety in how those motors are designed than just the two endpoints that I described here.
I see, thank you.
And the next question is by Kathleen Hall.
Yeah, I can. So we seem to have a lot of misfolded proteins or things that require chaperones. What do you think would happen if everything were simple and it didn't require refolding or helping, or if everything were simple? Would it be much faster?
Sure. Yeah, so
why is there so much help needed? I mean, evolutionarily, I mean, there's no real answer to that, but it seems like an awful lot of proteins need help.
Right, an awful lot of proteins need help. So let's see, I think there's a lot that could be said about this. First of all, there's a lot of proteins that don't need help. So if you look at E. coli, there's something like 80% of the proteins are very happy just blazing and to fold all the way up without any chaperones at all. And so that's pretty cool. But then the proteins that do need it really need it. And so the thing is, in one of the last slides I showed, if I had no chaperones at all, I have big problems because because the stabilities of proteins are so close to very small KT, a few KT. Protein stabilities are typically sort of 3 to 6 kilocalories per mole. And the difficulty with that is I'm going to get fluctuations all the time to unfolded states. And so not only do I have this, my whole proteome, let's say 4,000 proteins in E. coli, my whole proteome, I have lots of them that are pretty unstable. I think the average is about 6 kcal. So those are mostly okay, but there's a distribution function and there are lots of proteins, something like 10% of E. coli's proteins are only 3 kcals per mole stable. And those are all like lemmings just about to fall off the cliff. And you need something to help them. They're so marginally stable that they're fluctuating open all the time. And I just need a protection system around, especially when I'm now going to face any kind of stress. It's going to have to be able to function in stress, but it's also going to have to be able to function under normal conditions to keep the proteins folded. So the problem would be, and I think there's a very interesting deep question here in your question, which is timescales. So because if I just made a bunch of beautiful proteins, pristine proteins, and hands off, no proteostasis, no chaperones, and so forth, then it's going to degrade over time. And the question is, what's the time constant of that? We've looked at a little bit at certain time constants, and it turns out to be kind of remarkable. This 20-minute duplication time in E. coli happens to sit right between two other time-consuming activities. On the one hand, proteins all have to fold. And if I look at the distribution of lengths of proteins, my longest proteins fold pretty slowly, and they actually run up in the few minutes time scale to fold. So if I was to try to duplicate too fast, the trouble is I'm going to be duplicating proteins that aren't even folded yet. That's one problem. That's the problem on one side on the time scale. And the problem on the other side is that hydrolysis, protein hydrolysis and degradation takes place also sort of in the tens of minutes to days time scale. And so the thing is, you sandwich sort of right in the middle, that E. coli happens to grow right between sort of the two devils that it sees on the two sides. I got to fold my proteins, but I also don't want to degrade. And so I think what the chaperones do, if you want to think in terms of timescales, is it stretches out the permissible timescales. It stretches out how long proteins can survive and be able to allow you some plasticity and time scale so that E. coli can do whatever it needs to on whatever time scale. That's a guess.
Right. So there is another question by Catherine, but Catherine, I would like to shift you a little bit because I would like to give other people a chance who haven't asked a question yet. So next question would be by Ben.
Thank you very much, Ken. It's really very impressive how you're able to integrate all of this information and come up with these beautiful models. I was particularly impressed how well you were able to describe the temperature dependence of the age span of C. elegans. And so I was wondering whether you could tell us a little more, which aspects do you need to get right for this fit to work? Are there many different possibilities that would give you a right fit, or are there some key aspects that you need to get right?
Yeah, so that's a great question. So it turns out that time scale is dictated by a couple of things. So, and it turns out there's a kind of a narrow band of time scales here. You know, if you're talking about sort of the 10 to 15 degree range of C. elegans dropping its lifespan down by 2 orders of magnitude. And the question is, okay, what's dominant over this 10 degree part and what's dominant over this 10 degree part and so forth. So you're dealing with very, very subtle temperature changes with respect to the physics of proteins. Because the physics of proteins, of course, the temperature scales we think about are degrees Kelvin, or, you know, so you're talking about pretty wide ranges of temperature dependencies. One dominant factor that comes in is the folding itself, the temperature dependence of folding. The folding temperature dependence is a little more subtle than sometimes than people think, in the sense that if I look at the delta G of folding, people tend to think protein folding is pretty temperature sensitive. That's true if you care about the entropy of folding. It's true if you care about the enthalpy of folding. But if you look at the delta G of folding, it's actually pretty flat as a function of temperature. It's kind of independent, but it is one factor. The other factor is the trafficking. So we had to make an estimate there for what that trafficking is. We know the trafficking rates from the experimental data that comes from this community. And so we just made some ballpark estimates that were sort of sensible estimates is all I can say for them. But you do get different things at different points. And actually, let's see, I think I saw Adam on here. Adam, do you have anything you want to add to that?
Hi there. Yeah, if I remember correctly, the two main parameters are basically what is the fraction of your chaperones that are empty at 20 Celsius? Because most of the things in the cell are going to accelerate with Arrhenius type doublings, doubling every 7 degrees, 8 degrees Celsius. But the unfolding rate actually rises much more steeply. Every 2 or 3 or 4 Celsius it doubles. Okay. And so basically the reason why, humans can only endure a certain temperature raise before the fever becomes deadly is a little bit of the same thing that's going on here. And so we took a ballpark from the literature for what fraction of the chaperones are empty. And that basically tells you how many degrees Celsius you are from the cliff. And there's, the exact shape of the cliff. It's a pretty steep cliff. And so I wouldn't read too much into that. So, yeah.
Thank you very much.
Okay, so next question by Bingsu.
Great talk, Dr. Dill. I have a question about the, look at the healthy proteolysis in the cell. Is there any way we can think qualitatively whether, you mentioned in your slides that there are multiple, major ways to maintain healthy proteostasis, such as make new protein or repair misfolded protein or degrade misfolded protein. Is there any qualitatively we can think about which one is the most energy efficient, when you maintain those pathways, when we think about how we approach therapeutics for treat protein misfolding diseases?
So let's see. On the one hand, there's the issue of energy efficiency that you raised. On the other hand, there's, first of all, I don't know. We haven't calculated anything like a serious energy efficiency in the chaperone problem. The chaperone problem, the proteostasis problem, is already a sufficiently complicated set of ODEs, coupled ODEs, for us not to be able, I don't think I see a good way for us to get a handle on energy efficiency per se. The energy efficiency that I referred to there is a very coarse qualitative thing, which is to say, what I'm referring to as energy efficiency in the proteostasis part of the talk is simply that I can tell by how misfolded a protein will be based on which of the Ricardo classes it falls in. Some proteins are pretty sick, they misfold, they stay misfolded a long time. I can tell from that how long it stays misfolded, how sick it is, or I'm going to define that as how sick it is. Now, those tend to go to the most biggest energy users. So the GROEL is the biggest energy user. It uses a lot of ATP per every event, whereas DNA-KJ uses less and trigger factor uses none. So I mean energy efficiency in that sense. Now, I can't right now, I don't know how to go back to sort of a per ATP basis and compute it in any more granular way? I think it's a super interesting question, by the way, and it is something that we do care about. There's a lot of issues about energy efficiency. We interact a lot with Terry Waugh about this, by the way. There's a lot of numbers in E. coli, even just counting up ATPs. There's a lot of dark matter. We can't account for a whole lot of where energy is going and why wheels are being spun in the ways they are and so forth. There's a huge field in front of us of those kinds of problems to tackle. Anyway, so long story short, I can't tell you energy efficiency, but let me tell you one other related thing, and that is, which I think is part of what your question was aimed at, is suppose I could upregulate chaperones or downregulate them, let's say in certain diseases. How would I choose to make that decision? So there's a business out there, people who do a little of that. There's a drug that went out on the market several years ago called 17-AAG, based on the following idea. In cancer cells, I have a lot of broken, crappy proteins. So what the cancer cell does to respond to that is it upregulates its own chaperones. And because it's trying to fix what it thinks are broken, but they're too broken. So it's spinning its wheels, but it keeps cranking its chaperones up, spinning its wheels more and more. So here was the idea behind the drug discovery. And that was, suppose I could make a drug that simply shuts down or reduces the level of the chaperones. Gee, maybe I could kill the cancer cells because the cancer is trying to get its chaperones high. I'm going to drive them back down. So that's what 17-AAG did on HSP90. Now, what they found is that for certain cancers, it actually did work. But for many cancers, it doesn't, and ultimately got taken totally off the market. And I think the company went belly up simply because the side effects are horrendous. You can imagine the problem here. I'm dealing with a system that basically reaches to every molecule in the cell and screws it up in some way or another. And so this has big impact. If you screw up a chaperone system, it's going to have big impact across the cell in a lot of ways that are not understood at all. So it's complicated. And I know that there are now follow-on companies that are trying to do something and try to fix the sort of subtle side effects and so on. And, you know, I don't know. I wish them luck.
Thank you.
The second last question by Nikita Gupta.
Hello, that was a great talk. Thank you so much. I had a question about your minimal aging model. And I was wondering how changing the number of players or the steps in your model would affect the lifespan. For example, if you had more chaperone systems or more steps in folding, then it would seem logical that if it affects how fast the oxidation process happens. And so with this, can we make comments on systems that are more complex and more complex organisms, for example?
Okay, so now if I understand your question, you're asking about when we go to aging and when we now include our oxidative irreversible damage plane, we have done some reductions in how we think about the whole chaperone system. And you're asking how sensitive is that to the subtle distinctions that we make there? Is that your question?
I'm asking how, if you included more steps in the process, your model, the predictions of your model would change. If you had more steps in folding, or if you had more chaperone systems, how would your model change? If that makes any sense.
So here again, I think I want to pass this one to Adam. He's the guy who's got the insights into that. But what I would say is, I do think that Adam captured quite well the simplicity that was needed there to be able to get to us the sort of the minimal level we needed to be able to see the aging related phenomena we were looking at. Now, it depends very much on what property you care about. And if you cared about something that was very subtle, like this class of proteins or that class of proteins or this chaperone system or that one in aging per se, which is something that Adam is following up on. But if you cared about those level of issues, then you're at a level of granularity below the existing model, and you would then need to include those. Adam, do you want to say more about this?
Okay, yeah. I mean, I guess just to summarize that, I think there, so Susan Lindquist has some really beautiful work from the 90s and the 2000s that looked at this idea of the phenotypic capacitor. right? Where you can basically hide the frailty of certain phenotypic point mutations by having an overabundance of chaperones, right? But I think it's a double-edged sword. You don't want to have so many chaperones that you're buffering against even the accumulation of protein damage, for example, which is going to be a destabilizer of your proteins. And so I think evolution has to trade off between, how much cellular and protein heterogeneity are you willing to expose for the sake of selective pressure against those things, right? To keep the cell population healthy, and the robustness at the single unit level. Yeah, I'm not sure if that really gets to it. Like we would all love to be 100% folded, have absolutely no free radicals in the cell. But they have roles in signaling and feedback loops, and trying to understand this trade-off, I think, is integral. So
for context, my question came from this sort of, so the dream scenario would be to be able to predict the lifespans for more and more complex organisms. And I thought that if organisms were more and more complex, they would have more players in the system, which is why I asked this question. But thank you.
I'm sorry. Yeah, I think I interpreted the question how I wanted it to. Yeah. Email me, I can answer. Yeah.
Okay.
But one thing I would say here is that if you go to more complex organisms, then you do end up with huge numbers of chaperone systems and very little data. So the thing is, what we've tried to stick to in our modeling here, both in this proteostasis aging model and all of the other ones, is places where there's pretty good biophysical data that allows us to discriminate features of models, meaning what degrees of freedom matter, what are the fitness functions that matter, and things of that type. And I think at the moment, we would not be able to really undertake a project that says, hey, if we want to look at humans, for example, I think we couldn't do that just because the kind of data that we would need to have to be able to go to a lot more parameters would make it unpalatable.
Thank you.
So the last question is by Catherine.
So Ken, great talk. Thank you very much. You always make me think deeply. So I'm going to ask you to think deeply. I'm going to go back to one of your earlier slides where you said you pick your perturbation, your environmental change, then you form a hypothesis about how you think the proteome responds. So given that over 90% of the planet's microbial biomass exists at pressure,
yes.
That's what I would pick as the environmental factor. And do you have a hypothesis about how the proteome would respond?
We do not. We have not looked at that at all. And obviously, Kathy, that is a great question. And I agree, it's very important. And maybe we should communicate a little more offline about this. I know that there is some pretty good data, not only from you, but I remember the geophysical guys, for example, often have just a ton of data on this. And I guess we haven't really dug in enough to think about it, but maybe we should. I'd love to.
Thanks.
Okay, I think we had a pretty long discussion already, half an hour, and I think we should stop here. So thanks so much, Ken, for the exciting seminar, for the vivid discussion, and I hope I see you all in 2 weeks for the talk by Louis K. At the same time. And by this, yes, have a great day or great evening, and thanks again, Ken.
Thank you all very much.